Good morning friends. Today we learn about Histology of Spleen from B.SC. third year class. Paper number 2nd, Mammalian Histology. Spleen is the largest lymphatic organ in the body, located on the left side of the abdominal cavity. It is found between the stomach and diaphragm. It is soft, dark, purple and highly vascular organ. In this spleen, it also has a hilum. In spleen, here in the middle region, the hilum is present, and through the hilum, the splenic artery enters in the spleen. This spleen has hilum, which is where the major blood vessels enter and leave. Except at the hilum region, the spleen is covered by a layer. It is called as a peritoneum layer, and this spleen is covered by serous membrane called as peritoneum. The splenic arteries, nerves enter from the hilum region, and veins and lymphatic vessels leave the spleen at the hilum region. Beneath the peritoneum is a capsule of fibroelastic connective tissue containing some smooth muscle fibers. From the capsule, dense connective tissue forms the projection, extends into the substance of the spleen called as trabeculae. Here this capsule sends their septa, and this septa is called as trabeculae. These trabeculae divides the spleen area into different areas containing generally smooth muscles. This abundance of wavy collagenous fibers, elastic fibers, smooth muscle fibers of the frameworks enables the spleen to increase the volume whenever necessary. The space within the connective tissue framework are filled with the soft spongy tissue called as a splenic pulp. Histology of spleen mainly consists two parts, that is parenchyma and stroma part. Parenchyma consists splenic pulp, and this splenic pulp again divided into red and white pulp. These parenchyma mainly consists splenic pulp, and this splenic pulp divided into red pulp and white pulp. Red and white pulp. And second part is stroma. These stroma mainly consist myofibroblast cells, that is reticular connective tissue present in the stroma. Let us start with the parenchyma cells. These parenchyma mainly consist splenic pulp, and this splenic pulp, on the basis of color, the pulp is divided as white and red pulp. On the basis of color, these are divided into red pulp and white pulp. These both red and white pulp contains lymphoid tissues, that is reticular connective tissue. Lymphocytes, fixed pre-macrophages and monocytes are present in both red pulp and white pulp. Let us start with white pulp. This white pulp is compact mass of lymphocytes tissue around the arteries in the form of periarterial sheath. At the certain places, the sheath enlarges, and this enlarges in diameter, known as lymphatic nodules. It is also called as a splenic nodules. This enlarges called as lymphatic nodules, splenic nodules. It is also called as Malpighian corpuscles. White pulp contains framework of reticular fibers, lymphocytes, plasma cells and macrophages. In the white pulp towards the one side of artery there is germinal center present. In this white pulp germinal center is present where active multiplication of lymphocytes occurs. In this germinal center active multiplication of lymphocytes occurs. The germinal centers are numerous in the spleen of children but less numerous in the adult spleen. Next type is red pulp. The red pulp consists of plexus of veins, venous sinuses and which breaks into the cord called as pulp cord. It is also called as cords of bilirubin. It is also called as cords of bilirubin. This red pulp breaks into the cords called as pulp cords or cords of bilirubin. Red pulp also contains framework of reticular fibers, lymphocytes, erythrocytes, macrophages and the large number of erythrocytes in the sinusoidal which gives color to the red pulp. Next part is blood vessels present in the spleen. In the spleen, through the phylum, splenic arteries enter into the spleen and these arteries divided into branches which enter the arteries into the trabeculae to becomes trabecular or interlobular arteries. These, after short distance in the trabeculae, they follows surrounded by white pulp. The arteries becomes central artery. Here these central arteries present and these central arteries again follows separate course in the splenic pulp. Arteries get surrounded by white pulp. The arteries becomes central arteries and forms numerous capillaries which nourish the lymphatic tissue. Then their size is reduced and they lose surrounding coat of compact lymphoid tissue and enters in the cords of red pulp. Enters into the cords of the red pulp and the venous sinuses forms an plexus throughout the red pulp. When these pulp veins again forms collecting venules, they becomes trabeculae vein in the trabeculae and unite to form the splenic vein, and through the hilum the splenic vein leaves the spleen. Functions of the spleen: mainly the spleen acts as a filter organ for the blood. The phagocytic cells remove foreign particles such as bacteria and degenerating leukocytes. It destroys worn out RBCs and returns the HB to the blood, which is used to new RBC formation. Splenic functions as a organ of blood development during the fetal life, but after birth it forms only lymphocytes. After birth it forms only lymphocytes and monocytes. White pulp forms the antibodies. In the spleen, white pulp formation of antibodies takes place. In some mammals, it Spleen, it serves as a reservoir of the erythrocytes and blood. This is all about the histology of spleen. Thank you.